get to hear me talk all over again, but this time about early Earth geobiology, the topic of today's session. So, first off, I want to talk about what we mean by the early Earth. So this is a diagram of the timescale of the entire Earth, everything that's ever happened. And when we say early Earth in geology and geobiology, what we really mean is pretty much everything that happens from here, where the Earth is formed, really right up to about here, which is where multicellular life really starts to take off. So you can also think of it as everything that happens before the Great Oxidation Event in the Archean and Hadean, but everything that happens in the Proterozoic is generally included too. So essentially, the early Earth geobiology, that sounds like not a lot of Earth's history, right? But we're actually thinking about 3.5 or 4 billion years of the Earth's 4.5 billion year history. So this is clearly a lot of territory to cover. So what about geobiology? Well, in its simplest form, geobiology is where geology meets biology. In terms of geology, what we do is we look at rocks and we want to know what can they tell us about life in the past. So this picture up here, this is a picture of a formation known as the Gunflint Shirt, and it's really important. It was discovered in the 60s because all these things here, you think, wow, that really looks like a close-up microscope photo of a microbial mat. And you'd be right, this is a 1.9 billion year old microbial mat. So right away, this rock is telling us something about the organisms that were living 1.9 billion years ago. But there's another part to geobiology, and that is asking what life in the present can tell us about the rock record. So this picture, this is of a stromatolite, which some of you have probably heard of. They're structures found in the rock record that we think indicate microbial life. But this stromatolite is really cool because this stromatolite isn't 2 billion years old. This stromatolite was found in a Yellowstone hot spring about 5-6 years ago, but it looks exactly like a whole bunch of stromatolites from the rock record. So by studying things like this that exist in the present day, we can understand what the structures in the rock record actually are and what might have been going on back then. So let's just go back to this kind of diagram of the timescale of the Earth for a minute, and I want to go through some of the key events in early Earth history to give you a good idea of what was going on at different times and the kinds of topics that our speakers today will be talking about. So we really kick off in terms of geobiology roughly 4 billion years ago, which is when the oldest known rocks that we can access were being formed. So anything that happened before 4 billion years ago, we don't know about from the rock record on Earth because those rocks aren't there anymore, they're gone. About 3.5 billion years ago are the oldest stromatolites, the oldest probably microbially formed structures. So that's the first point at which we can look at these things and say this is probably life, and we're seeing evidence of it in the rock record as a structure, just not in terms of our predictions. About 2.3 billion years ago came the Great Oxygenation Event, and this is when we can say, okay, there's not just life on the planet, it's creating so much oxygen that it's getting into the atmosphere and fundamentally changing how our planet is. This is the point at which the early Earth changes from being a primarily anaerobic world to a not as aerobic world as today, as we were told about last night, but still a world where oxygen is playing a major chemical role. So this is a really crucial changeover in our early Earth time period. About 1.9 billion years ago, that's the Gunflint Shirt I was showing you. This is when we have the oldest microfossils, so we can zoom in on a rock and actually see organisms preserved in the rock. It's also when we have the oldest eukaryote fossils, so we don't just have prokaryotes, we've got eukaryotes and we have evidence of them. About 1.2 billion years ago, we have the oldest identifiable fossils. And what I mean by that are fossils where we can look at them and say, in the case of these particular fossils, these are red algae. So we're not just saying these are microorganisms, these are members of a clade that exists today that we know about. About half a billion years ago, so about 600 million years ago, we have the oldest multicellular fossils. And I'm going to zoom in a bit on the timescale just to bring you up to speed on where we hit the modern period. And so this is really, really zoomed in, but this is everything going back to 600 billion years. So this is the dinosaurs, this is us, and it's a tiny fraction of the whole history of the Earth. So about 600 million years ago, we have sponge fossils. But when you fossilize sponges, what you mostly get are these tiny little silica glass spicules. So they don't look like a whole sponge, but the evidence that silica-forming sponges were around. About 550 million years ago, maybe 600, you get small shelly fossils, and these are the first organisms that were forming actual shells. And then you get the Cambrian explosion, or the major animal phyla appear in the rock record, and we're off to the races with multicellular life. But what we really care about is the first 3.5, 4 billion years before that, when multicellular life wasn't around. So when you don't have fossils in the rock record, when you don't have your bones and your coprolites and your trace fossils, what are you looking at to tell what was going on with life? So back to our diagram. You'll note that on this diagram, the Hadean and the Archean, the early anaerobic period, are in red and pink. And this is important because I'm going to show you this map. And the Archean rocks, which also encompasses the Hadean for these purposes, also red and pink. So right away you can see when we go looking for rocks to find out about life in the early Earth, we don't have a lot of rocks to go look at because not all of these are going to tell us about life. Life is primarily preserved in the sedimentary rock record, not the igneous or the metamorphic, because that's where life tends to live. Life isn't living in basalts and volcanoes, it's living in the soil, it's living in the seafloor. So we have to find sedimentary rocks, and there just aren't that many of them. Apart from the very top of North America, most people also study Western Australia, South Africa, and there are Archean rocks in India, but not a lot was done with them. So there's really only these 2 or 3 areas in the world where you can go to find these rocks and find out about the early Earth. So we have to work really hard with what we've got. So what are we looking for in these rocks, since we don't have the bones and the coprolites and all that sort of thing? And we're looking for evidence that life was there, which can be microfossils, although again, we don't have a lot of microfossils for the early Earth. They start to emerge sort of 2 billion years ago, really post-Great Oxidation event. We're looking for evidence that life could have been there. We're looking for evidence that life could not have been there. And if this is starting to sound like astrobiology, you'd be right, because this is the thing about early Earth geobiology. For us, the early Earth is basically an alien planet. It's this anaerobic world filled with prokaryotes where life as we know it doesn't really exist. And the only way we can look at it is to study rocks, and there's only a very few of these rocks because most of them have been destroyed. So it's pretty much exactly like looking for life on Mars. You're looking at the few rocks you can get hold of for a totally alien world. So this is early Earth geobiology isn't just important because it tells us things about what alien worlds might be like. It is telling us about an alien world, the beginning of our own. So these are some of the kinds of things you look at when you're looking for evidence in the rocks. Microbial structures, so not necessarily microfossils where you're looking them under a microscope and seeing the actual microbes, but structures created by microbes. And some of you who are not geologists are probably looking at that and thinking the only structure I see there is a rock hammer, and that's fair enough. But this is a stromatolite. The term stromatolite actually means a mattress rock because you have layers like mattresses that create this structure. They don't look a lot like mattresses to me, but clearly they look like mattresses to someone, so that's what we're going with. And if we zoom in close, this is a pre-Cambrian, so not like super, super early Earth, but like around the time of or just before the Cambrian explosion, stromatolite. It's at Mount Dunphy in Nevada. I was there a couple of months ago where I took this photo. And you can see that there's all these fine layers in this fine-grained sandstone, and we think these are being caused by little microbes growing in mounds, and then they're trapping sediment somehow, and it forms layers, and these are then preserved in the rock record. You also get, going back to the gunflint chert, actual microfossils, normally not for the very, very early Earth. There are some examples like the apex chert, which we mentioned last night, which are very hotly debated, because they're kind of like the Martian meteorite found in Antarctica. They look like microbes, but it's so long ago and the rock's so screwed up that are they microbes? Maybe, maybe not, but these definitely are. So for the later early Earth, we're really lucky we can actually see microbes in the rock. But without these larger microbial structures and the microfossils, what are we left with for these really old rocks that we don't have many of? Well, there's a few proxies we can look at to think about life too, and you'll hear about some of them today. There's isotopes, which you're going to hear a lot more about from Bradley in just a few minutes and from speakers later on this afternoon. But essentially, isotopes like carbon, oxygen, and sulfur can tell us things about the chemistry of the early Earth, which can give us ideas about what sort of life could have been there, and especially in terms of oxygen, whether life was producing oxygen and what it was interacting with. You have lipids and organic molecules. Things like DNA don't survive that long. The oldest ancient DNA that we've managed to sequence is maybe 30, 40, 50,000 years old. There's claims of older stuff, but it's pretty broken up. But things like lipids can survive a really, really long time. Again, this isn't really relevant for the early Earth, and the best examples are actually probably from the Phanerozoic, which is the modern Earth post the Cambrian explosion, but you can get the preservation of lipids in the rock record, and you can look at the lipids and think about the kind of things in the modern world that have those lipids in their membranes, and so what it could mean about the life that could have been there. And finally, and key for geobiology, there are minerals. Hooray. So this is an example of a banded iron formation, and I'm sure a lot of you have heard of these. This is where in the around the time of the Great Oxidation Event, you had tons and tons of iron being laid down as the iron rusted with oxygen and was laid down in these beautiful red layers. And so these minerals are telling us again something about life on the early Earth, because they're telling us about the oxygen. And you get other minerals. You get iron minerals that could be created by microbes that reduce and oxidize iron. You get minerals like sulfur minerals, some of which are biogenic. Later on in the Earth's history, you get carbonate minerals by organisms that are forming carbonate, either directly or by their influence on the environment. So rocks are affected by life. It's geobiology because the biology can create the geology. It's not just coexisting with it. And furthermore, in early Earth geobiology, we also have modern-day analogs. So there's places like deep-sea hydrothermal vents, which are very hot, they're anaerobic, and organisms are existing with the rock. There's no sunlight, and they're creating their own biosphere down there. You have places like Yellowstone, where you've got these hot springs, and stromatolites, just like the ones we see in the rock record, are being created right now by organisms existing in the present day. You have places like the Black Sea, where you have a huge anoxic basin in this really quite large sea, where the ocean is anaerobic, like we think it was in the early Earth. Now, it's not a perfect analog because the concentrations of a lot of things like sulfur are very different, but it's still a whole anaerobic section of ocean that we can go look at to see how organisms are getting on, because there ain't no fish down there, it's just microbes, just like we think it was on the early Earth. But really the point of all this for us as astrobiologists is that if we know what life looks like in the rock record here and the signs it leaves behind, we can understand what it might leave behind on other planets and how we can really adequately go out to Mars and look at the rocks and say, this is geobiology. These are the signs of what life left behind there, just like it did here. Thank you.
So I'm going to be rude and take the first question myself. I was intrigued by the rock distribution map that you put up at the beginning.
Yep.
What is it about northern North America that is the sole source or primary source of that sedimentary stuff? Is it glaciers? Or?
So it's not the primary source. Western Australia and South Africa are a little better. That was the best picture I had. But the thing about North America is you have this sort of craton, which is the center of North America, and it's like the center of a continent that's had stuff added onto it and stuff broken off at the edges, but the center is really old. And that's why you have all these old rocks preserved.
Any other questions?
Away from you as possible. Yep.
So you showed this picture of layered rock, and you said that you think his layers are formed by bacteria. Can you explain how you know that these layers are formed by bacteria?
So that's a great question, because the original thinking was that all stromatolites were formed by bacteria, and there's a growing idea that actually a lot of stromatolitic structures aren't necessarily microbial. How you can usually tell is you have layers and they're laid down flat, but the flat is along a cone shape, and the grains are trapped in ways where if it was just dropping on there, they would have rolled off if they weren't being trapped by something sticky. And we think the most likely thing in those situations is there were organisms, they were producing sticky extrapolyar saccharides, or they were actually filamentous growing around the grains, and that's why they're being trapped. You can certainly have structures that look like that, but if you look at the microstructure, it's hard to think of ways that biology could have been involved. So there's definitely a range. Not all stromatolites are definitely biological, but a lot are.
All right, we need to move on, so let's thank Lucy again.